with the gel electrophoresis. अच्छा जी जेल इलेक्ट्रोफोरेसिस का जो प्रोसेस है माय डियर दैट resemble with that of the paper electrophoresis, but the only difference lies the results of the paper they are only visible for 1 or 2 days. Okay, after 1 or 2 days the results of the paper electrophoresis they vanish. Okay, so you have to do again the experiment, okay, in order to get the result. But gel electrophoresis, it got the results for a longer period of time. Okay, so for that purpose, what should you do? You should take a piece of glass like that or piece of plastic. So that's your choice. Okay, so you should take a piece of glass or piece of paper, a piece of glass, sorry, or piece of plastic. So normally we take piece of glass. Glass is more good. Plastic we don't take normally. Okay, but you can take the plastic. Why? We don't take the plastic because in Pakistan, whatever the gel we use, okay, that gel basically is not giving good result on the plastic. On glass, it is glass surface that is slippery, so it will give you the perfect result. Okay, so that is why I use the glass. I do suggest you. Okay. Now, my dear students, what should you do? You should put some pieces of paper or pieces of thread up here, like that. Okay? Pieces of paper or pieces of thread. So, let me write this thing up here. So, this is the piece of glass. Okay, now you should put these pieces of thread, okay, on the glass slide, okay, and dip it into a buffer solution. Okay, so this is the buffer solution of pH 9, and this one is dipped into a buffer solution. Okay, so this is the buffer solution of pH 8 to 9. So this one is also 8 to 9. Okay, so basic pH we have taken. Okay, so now what would happen, guys? This buffer solution that is going to be absorbed by this thread. So this is the thread or the piece of paper. Piece of paper और इस piece of paper को आप लोगों ने what you have done, you cut it into a small piece and put that small piece of paper from there into the buffer solution. From here into the buffer solution. Okay. Now this buffer solution that is going to rise on the surface of this thread or the piece of paper. Okay, guys. So that piece of paper or the thread is present like that. Now, what should you do? You should apply a gel on its surface like that. So, why you put this thread or the piece of paper up here? What is the reason behind it? I'm going to tell you about it a little bit later. Okay. So, anyways, you have applied the gel up here. So, this is the gel you have applied. Okay. Now, you know that the gel surface that is slippery. Okay, so if you drop the buffer solution from the top, it will slip on the surface of the gel and will go down. So it would not be absorbed by the gel. So that is why, my dear students, we put the pieces of paper or the thread up here and dip it into the buffer solution of pH 8 to 9. Okay. Now, this gel that is actually the polyacrylamide gel, or this one that is also called as the agarosol. Okay, so this is the agarosol, or this one that is also called as the polyacrylamide gel. Okay, Uh, so you put the gel up here, okay? So now your instrument that is ready to work, okay? Now what should you do? You should take a knife and put a well inside it, okay? Inside the gel, okay? And now put the mixture. You should put the mixture of amino acids with nin hydrogen in it. Okay. So nin hydrogen will make these amino acids as the purple. Okay. Because when the amino acids they would move here and there. Okay. So basically they won't be visible. You know that amino acids they form the colorless support. Okay. So if you put the nin hydrogen with them, so they would become colored. Okay. So when they start moving, so the purple spot formed at any place that will tell you the place of that particular amino acid at that place. Okay, did you get it?
Yes, sir.
Now the rate of movement of these amino acids here you apply the electric field like that. Okay, this is the electric field you applied up here. So this is so apply the positive. This is the negative. What would happen up here? So you just dug up the well, little bit well up here. You put the mixture of amino acids and main hydrogen up here. You apply the current. So this is the positive terminal. This is the negative terminal. I told you that the buffer solution that is spread within the gel, okay, due to these threads or paper, pieces of paper, okay. So the amino acids in that particular buffer solution, they would become positively charged, they would become neutral, they would become negatively charged, okay. So now they will move at different sides, okay, and the rate of movement I told you yesterday, it depends on basically the charge on the amino acid. The second thing, amount of current applied, and the third thing that is the MR of amino acid. Okay. So now write down these things up here. The first thing, take a glass slide, put the two pieces of thread on it and dip them in the buffer solution of pH 8 to 9. Okay. Now the next thing, my dear, apply apply the polyacrylamide gel or aerosol on the surface of glass slide. Let it dry. Okay. Now dug a hole in the gel by means of a knife and mixture of amino acids and hydrogen in this well. Apply the high voltage electric current then amino acids in the given pH would be negatively charged, positively charged, or neutral. Neutral means in the form of sweeter ion. Sweeter ion, I told you, they are the neutral ones. They got positive and negative on both ends, so that is why they are neutral. Okay, so amino acids they would change into negatively charged, positively charged, or neutral sweeter ions. Okay, uh, different ions will travel on different sides the rate of movement of ions depends on the current applied. Okay. The rate of movement of ions depend on number one, the AR or MR of amino acids. Okay, number two, the charge on amino acids. And the third thing, current applied. Okay, my dear students, now the next thing, so amino acids are separated and can be identified by comparing नहीं या comparing their distance traveled with the standard amino acids under same conditions. What we had did before, हम लोगों ने पहले क्या किया होता है that we had done the electrophoresis of every single amino acid like glycine, alanine, phenylalanine, glutamic acid, हर एक का हमने करके रखा होता है कि वो किस तरफ को ट्रेवल करता है और कितना ट्रेवल करता है, वो डिस्टेंस हम लोगों ने मेजर किया होता है. Now what we have to do when we got the mixture of amino acid, तो उसमें different spores बनेंगे. उन spores का distance central point से आप लोग measure कर लें जहां पे बेल बनाया हुआ है. And when you measure the distance, so compare it with the standards you have made for different amino acids under the same conditions. तो उससे आप लोगों के पास different amino acids identify हो जाते हैं. Did you get it?
Yes, sir.
With the DNA fingerprinting, that is a very important topic. DNA fingerprinting. DNA fingerprinting. This is the application of this gel electrophoresis. Okay, ये थोड़ी सी मुश्किल है, जरा प्लीज ध्यान से जो है वो सुन लीजिएगा. Little bit difficult, but this is the application of gel electrophoresis. Okay, so how could we do the DNA fingerprinting? So let me describe this thing for you. Okay, basically DNA fingerprinting in our countries that is basically used to establish the relationship, okay, between any child and his mother or his father, ठीक है जी. uh, और इफ यू पीपल हैव टू गो टू द अमेरिका, वहां पे आपके फादर या मदर है, तो यहाँ पे डीएनए फिंगरप्रिंटिंग करके चेक करते हैं कि बाकी ये उसका फादर है या बाकी इधर उधर उसकी मदर है जिसके पास नेशनैलिटी है, तो तब आपको जो है ना वहां की मतलब नेशनैलिटी वगैरह वो ऑफर करते हैं, तो डीएनए फिंगरप्रिंटिंग. लेकिन हमारे मुल्कों में तो इसका यूज जो है वो कम होता है, लेकिन अभी रिसेंटली जो है वो इन्होंने यूज करना शुरू कर दिया है डीएनए फिंगरप्रिंटिंग का. लेट्स सपोज गाइस, किसी जगह पे कोई चोरी होती है, बस हम सपोज करें कि दिस इज द रूम, अब इस रूम में चोरी हुई है, ठीक है. तो जो डाटा कलेक्ट करने वाले या सैंपल कलेक्ट करने वाले होते हैं वो आते हैं. अगर मैंने इस व्हाइट बोर्ड को टच भी किया तो यहाँ से मेरी स्किन के सेल जो है वो यहाँ पे लग सकती है. यहाँ सपोज करें कहीं पे कोई नेल लगा हुआ था और मेरा माय हैंड टच दैट नेल, तो ब्लड निकला है, तो ब्लड का ड्रॉपलेट दिख जाए, दैट इज आल्सो द सैंपल. ओके, और ये आपके जो बाल हैं इसमें से कोई एक हेयर पीस नीचे गिर जाए तो दैट इज आल्सो द सैंपल. ओके, सो इफ यू हैव जस्ट ड्रिंक द वाटर, तो आप लोग ये वाटर ड्रिंक करते हैं तो आपके जो चीक सेल्स हैं वो यहाँ पे लग सकते हैं, तो दैट इज आल्सो द सैंपल. तो जो डाटा सैंपल कलेक्ट करने वाले होते हैं वो क्या करते हैं कि डीएनए का सैंपल जो है वो सबसे पहले कलेक्ट कर लेते हैं, ठीक है. तो द फर्स्ट स्टेप इज टू कलेक्ट द डीएनए सैंपल फ्रॉम अ साइट. ओके, ये बाहर के मुल्कों में जो चोर पकड़े जाते हैं वो इसी तरीके से पकड़े जाते हैं, उसको टाइम लगता है. वो जरूरी ये नहीं करते कि वो सिर्फ और सिर्फ जो है वो रिकॉर्डिंग्स को देख के जो है ना वो उसकी पूरी उस प्लेस की डीएनए फिंगरप्रिंटिंग करके एक एक चीज का सैंपल एनालाइज करके चेक करते हैं कि क्या ये बंदा वाकई यहाँ पे आया था, वो सारा कुछ चेक करते हैं. तो फर्स्ट ऑफ ऑल टेक द सैंपल, and sample that should contain the piece of hair, that should contain the blood sample. Okay, it must contain the skin cells, it contains the cheek cells. ठीक है, तो कोई भी डाटा जो है वो आपके पास जो है वो हो सकता है, बॉडी का कोई भी पीस जो है वो किसी बंदे का, इफ यू गेट एनी पीस of the body of a person, from that part you can collect the DNA. Okay, because you know that living things they are made up of cell. I told you last time. और हर cell के अंदर nucleus got the chromosome, and chromosome that is made up of genes, and genes they are made up of DNA and protein. So DNA is present in every cell. Okay, in the nucleus. Now you take the sample first of all. Okay. The next step, you have to separate the DNA. Okay, so you have to add it into a machine. This machine is called as the centrifuging machine. Okay, so this centrifuging machine, my dear, what does it do? This is a machine. It got some particular speed. इसमें टेस्ट ट्यूब में सैंपल जो है वो डाल दिए जाते हैं, and this test tube, this machine that is going to uh, rotate the sample like that and revolve it like that at the same time. See, have a look up here. So this one is being done at a particular speed. We know that at particular speed only the DNA that would remain and the other things they will destroy. If you want to get any other material from the sample, let's suppose you want to collect the nucleus, whole nucleus, or you want to get the whole chromosome. So the speed that will be different for centrifuging machine. तो सेंट्रीफ्यूजिंग मशीन की स्पीड हमें पता होती है कि इसके ऊपर ये डीएनए का पीस जो है वो एक्सट्रैक्ट हो जाएगा. सो व्हाट वी डू बाय यूजिंग दिस मशीन, व्हाट वी डू, वी एक्सट्रैक्ट द डीएनए. यहां तक क्लियर हो गया गाइस? डिड यू गेट इट गाइस? प्लीज टॉक टू मी. आई एम
Yes,
आस्किंग.
sir.
यू आर सोइंग सारे यार. एक्सट्रैक्ट द डीएनए फ्रॉम सैंपल. एडम, you should talk. But sir, you should talk. Muskan, you should talk, beta. You should give me the answer.
Yes, sir. Got it.
Thank you very much. Okay, extract the DNA from the sample. Okay, now what should you do? You should add the restriction enzymes in the sample of DNA. I told you last time that the DNA that is actually like that. Okay. So now what the restriction enzymes? Restriction enzymes you can understand that basically these are the scissors. Okay, so what they do, they cut the DNA at different places and divide this DNA in different parts. Some parts are big, some are small, like that. Okay, so different fragments, different pieces of the DNA that is being formed by this restriction enzyme. So restriction enzymes, they are basically the scissors. They are just like the scissor. They cut the DNA. So restriction enzyme, what they do, they cut restriction enzymes cut DNA at different places and small and large pieces of DNA are formed. Okay. Now, my dear, what you have to do? These large and small pieces of the DNA, we have to do the process of electrophoresis for them. So their electrophoresis, DNA electrophoresis, that is little bit different. Okay, so we will do that electrophoresis inshallah tomorrow, and tomorrow this topic of electrophoresis that will be completed. Okay, so that's it for today from my side.